Hello guys, I'm Fredrick. I'm in Form 2. Let's share something important about cell and Form 1 work. At the end of this lesson, you will be able to know what's the meaning of cell, the function of microscope, type of microscope, difference between light and electron microscope, structure and function of light microscope. You will be able to calculate the magnification when using light microscope, and you will be able to know how to take care of light microscope. The cell is the basic functioning unit of the body. For example, this one is a cell. As you can see, this is an example of plant cell. It has different parts on it. The function of microscope is to magnify specimen and take resolution on it. Resolution is the ability of microscope to distinguish between two close structures. The difference between light microscope and electron microscope is that light microscope use light to illuminate specimen, but electron microscope use beam of electron to illuminate specimen. Specimen view under light microscope should be dead or alive, but specimen view under electron microscope must be dead. Light microscope has low magnification power, while electron microscope has high magnification power. Light microscope has low resolving power, but electron microscope has high resolving power. Light microscope uses glass lenses, but electronic microscope uses electromagnetic lenses. On light microscope, specimen is placed on an open stage, while on electronic microscope, specimen is placed in a vacuum. We use normal dyes when staining a specimen on a light microscope, but we use a complex dye when staining a specimen on an electron microscope. On structure and function of a light microscope, this is the picture of it. As you can see, the base is here, which provides firm and steady support. The mirror that reflects the amount of light passing through the condenser to the stage. It reflects the light passing through the condenser to the stage. The diaphragm regulates the amount of light passing through the condenser to the stage. Condenser concentrates light passing through it to the stage. A stage, which is a flat platform where a specimen on slide is placed. A clip that holds a slide on its position. An objective lens that brings image into focus and magnify it. A revolving nosepiece that enables the objective lenses to change from one to another and hold objective lenses. A body tube that hold eyepiece length and objective lenses. An eyepiece length that have lenses that enable one to magnify specimen. We can say it project image further. It magnifies image further and project it to the eye. A coarse adjustment knob that bring image into rough focus by raising it and lower it for a further distance or a longer distance. A fine adjustment knob that is being lowered and raised for a shorter distance to bring image into a fine sharp focus. An arm that holds a body tube and the stage. On how to calculate the magnification when using a light microscope, the magnification is taken as eyepiece length magnification times objective length magnification. For example, if eyepiece length is 10 and objective length is 45, then the answer is 450 after multiplying it. If you want to take the estimation of cell size, you should know how to take the diameter of one cell, and that is the diameter of field of view over the total number of cells. For example, if the diameter of field of view is 5 and the number of cells is 10, you should change the diameter of field of view into micrometer by multiplying it by 1000 and divide with 10. then the answer will be 500 micrometers. I want you to know if you want to calculate the actual size of a cell, you should take the estimated size over total magnification. On how to take care of a light microscope, I want you to know that the first thing, you should always carry a microscope with two hands when moving it from one place to another. You should use one hand to hold the arm of the microscope and another to hold the base of the microscope. Always place the microscope some distance away from the edge of the bench. Never touch the mirror and lenses with bare hands or fingers. Clean dirty lenses using special cleaning paper. Do not pour water on any part of the microscope. Ensure that low power objective lens click in position after use. Store the microscope away from dirty place, in a place where it's safe and free from moisture and even dust. Thank you. This is the only thing that I needed you to share with me, and let's meet together when talking more about cell organelle. Bye.